Okay, we're still talking about passive diffusion, but now we're talking about a type called facilitated diffusion. And so if you think about the word facilitated, that means easier or make it easier or to help with or something like that. And so in facilitated diffusion, transport proteins are going to go across, are going to speed the passive movement of molecules across the plasma membrane. And so they're going to do it in a couple of different ways that we're going to see. So remember back to our channel proteins where they provide corridors that allow just a specific molecule or ion to cross the membrane. And again, we had talked about aquaporins, and then we also have things that are called ion channels that will open or close in response to a stimulus. And so a lot of times it may be, some sort of ion. These are often called, these ion channels are also often called gated channels. And so here, up at the top, we have a channel protein here. Down here we have a carrier protein. And so the thing that I want you to notice here is that this is just a plain channel. There's no extra space in it. It's only going to allow one to go through at a time. In a row. But down here with the carrier proteins, you actually will have to bind into a special configuration in that carrier protein, and then it will close, and then it will open to the other side of the membrane and dump that protein, or excuse me, dump that molecule out. And then it will turn around to the other side of the membrane, open back up, accept a solute, close, and then open up to the bottom and drop it into the cytoplasmic membrane. So that's really interesting to see that. Facilitated diffusion is still passive because the solute is going to move down its concentration gradient. And so what we've been talking about so far is, the movement from a high concentration to a low concentration. But there are some transport proteins that will move the solutes against their concentration gradients. And so these are going to need a little bit of help in order to allow that to take place, to move against their concentration gradients, go from low to high. And so this is called active transport. These are going to move substances across against their concentration gradient. Again, this is going to go from low concentration to high concentration, just like passive is moving from high concentration to low concentration. But in order to do that, the active transport needs to have a little bit of energy, and that's usually in the form of ATP, which is, adenosine triphosphate, which is the power of a cell. Active transport is performed by very specific membranes, very specific proteins embedded in the membranes. So a lot of your carrier proteins are going to be able to, or are also going to be able to do active transport, but none of your channel proteins will be able to do this. Active transport allows cells to maintain concentration gradients that will differ from their surroundings. For example, the sodium-potassium pump is one type of active transport system. This is probably one of the most common or most known about. Another one would be ATP hydrolysis, where you basically remove a phosphate and attach it to a transport protein. And there are several other types of active transport out there, but like I said, they must use a little bit of ATP. And so here's an example of the sodium-potassium pump, and you'll notice that what we're going to do is we're going to move from a low sodium to a high sodium concentration. So we're going across, or basically up the concentration gradient, and in order for that to happen, a phosphate is going to have to be attached to this pump. And so here's this phosphate right here. And the ATP, basically it took one of those phosphates off and it became ADP. And what that allowed it to do is, you'll also notice that over here, the shape of the holes are for the sodiums are this nice round shape, but then over here where it's releasing the shape of releasing that sodium, the shapes become more narrow, and so there's no physical way that those sodiums could attach back into the active transport system. You'll also notice that over here for our potassium, it is more of a very slender triangle, and at this point on the left-hand side of the picture, there's no way that potassium could bind, because it needs a wider pyramid shaped in order to accept that potassium. So on the next page, we will see this is moving the sodium across from the cytoplasm to the extracellular fluid, where you have a higher concentration in that extracellular fluid. And here you're moving the potassium from the lower concentration on the outside, and so low potassium, and then you're going to move it to a high potassium. Okay, so here you can see that those potassiums are going to fit very nicely into those wider triangle shapes, and the protein will actually just kind of pivot and release those potassiums out into the cytoplasm. But again, in order for this to happen, you're going to have your phosphate attached for the sodium, and when the phosphate detaches, that's when the potassium sites change and allows those potassium to kind of pop out. So I don't know if this animation will work. Let's see.
The sodium-potassium pump is an active transport mechanism. 3 sodium ions bind to the protein channel, and an ATP provides the energy to change the shape of the channel that in turn drives the ions through the channel. 1 phosphate group from the ATP remains bound with the channel. The sodium ions are released on the other side of the membrane outside of the cell, and the new shape of the channel has a high affinity for potassium ions, and 2 of these ions now bind to the channel. This binding again causes a change in the shape of the protein channel, and this conformational change releases the phosphate group on the cytoplasm side. This release allows the channel to revert to its original shape, and as a result, the potassium ions are released inside the cell. In its original shape, the channel has a high affinity for sodium ions, and when these ions bind again, they initiate another cycle. The important characteristic of this pump is that both sodium and potassium ions are moving from areas of low concentration to areas of high concentration. In other words, each ion is moving against its concentration gradient. This type of movement can only be achieved by the constant expenditure of ATP energy.
Okay, so coming back to our slide. So he always makes it so much more sophisticated sounding than anything I could put into words. So that kind of gives you an idea. It's really the attachment of that phosphate from ATP that's going to allow the sites, the binding sites within that carrier protein to change shape, and that's where you're going to get the affinity, which means the attraction for either the sodium or the potassium. So again, we have our passive transports, either diffusion or facilitated diffusion. And then our active transport, and of course, the main thing about our active transport, again, is going to be that ATP lending its phosphate to the protein itself. I'm sorry about that scuffling you hear in the background. Those are my dogs. They can't seem to leave me alone. They don't want to, they don't want to get out of my room while I'm recording, so I apologize. All right, so small molecules in water are going to enter or leave the cell through the lipid bilayer or by transport proteins. So those are small molecules in water. Larger molecules, such as polysaccharides, so larger carbohydrates, and proteins cross the membrane in something called across the membrane in bulk via these things called vesicles. And this is also another one that requires energy. So this bulk transport is going to require energy. It's going to need some of that ATP. And so we're talking about exocytosis and endocytosis. And in exocytosis, the transport vesicles migrate to the membrane, fuse with it, and release their contents to the outside of the cell. And so a lot of your secretory cells are going to use exocytosis to export their products, such as insulin. Insulin is secreted by exocytosis into pancreatic cells. Oops, going back. Whoops. Into pancreatic. Oh my goodness, I don't know what I'm doing here. Okay. Into pancreatic cells. There you go. Okay, sorry about that. I had to deal with my dogs scuffling, and again, I apologize for that. All right, so remember that anytime you see exo, that means it's going to go out. Go out. So if you see exocytosis, that means you're going to it's going to be made within that cell, but it's going to be excreted outward. Okay, so if we go to the next one, which is endocytosis, so endo means in, so this means go in. And so this means that the cell is going to take in macromolecules by forming vesicles from the plasma membrane. Endocytosis is basically just the reverse of exocytosis, but it involves different proteins. Trying to think of, let me see. Okay. I'll try to find an animation that we can see this taking place. All right, so here are a couple of examples of phagocytosis. And so here is the material. Phago just means food. And so this is going to be an endocytosis. So we're going endo here. And so basically what's happening is here's the foodstuffs right there that the cell wants. They're going to produce this pseudopodium, which means false foot. So pseudopodium. Podium means false foot. And basically they're going to surround that foodstuffs and bring it into a vesicle that is completely enclosed by a bilayer membrane, which was the membrane of the cell. And basically the same thing over here. You see an amoeba engulfing a bacterium via phagocytosis. And so here, this right here, that is the false foot. And then this right here, this is the bacterium that it wants to engulf. And again, that's going to be endocytosis. Let me see if this animation works. No, okay. All right, let me see if I can get one that works. Amoeba endocytosis. Let's see if we can get something like this. All right, here we go. This is well, actually, let's look at this one right here. So this one right here, I'm not going to show it in its entirety, but definitely go and look at it if you have a chance. And so I'm going to turn this off so I can talk while it is. Hurry up. Come on, don't have time for this. Phagocytosing, basically, Here is your amoeba. And what's going to happen is this little guy is a paramecium, and the